What kind of thing?
By being brave to choose.
If you ask what we want it to be, we can choose a hard one.
Yeah.
So will your committee members just stare at you?
Yeah.
I hate that.
And maybe if there's nice weather outside, you might take lenses out there. For the most part, they're just gonna be looking at you again.
I hate that. Mine was over Zoom. I don't know.
Oh,
You guys
no, I got mine in person. I got mine in person. Mine was in this room.
Yeah.
So. Yeah, mine was in this room. This is a good room to do so.
I'm redrawing.
Waiting.
Is Bruce on your committee? Is Bruce on your committee?
No way.
Not yet, but I was gonna ask him.
Could you explain as you're going?
Okay. So I drew a tripartite glutamate synapse. We have the presynaptic terminal arising from the axon where action potentials are propagated. They hit the active zone of synapse, which facilitates vesicular release. Vesicles are loaded with glutamate from the vesicular glutamate transporter, released into the synaptic cleft, where they activate AMPA receptors as well as NMDA receptors. It's taken back up by both cell types as well as astrocytes by glutamate to be recycled and repackaged.
Are ionotropic receptors the only type of receptor?
No, there's also metabotropic
And
receptors.
where would those be?
Glue 5 and 3 are all perisynaptic, and I think the other ones are in the synapse. I know 5 is perisynaptic.
It can be on both pre and post.
Yeah.
Is that what you mean by peri?
No, I meant outside the
Oh,
synapse.
okay.
I need to think of another shape.
You can hug it, right?
No, this is
Can we ask questions? I don't know if this is important, but how is glutamate turned into glutamate? Does that make sense?
So it's, yeah, glutamine.
Where is that?
It's actually a complication.
Yeah.
Where
This
is
is-
it?
Yeah.
Yeah, if you guys do anything with
I thought that happened inside the body, but I'm not sure.
So it,
Isn't it acidic or like something?
yeah,
There is something
Being
that-
part, oh, go ahead.
can I go draw it?
Yeah.
Oh, cool.
Yeah, I know this is just.
Good job.
I have to reorient this in my head. So this is the three.
Oh,
Glutamine is remained in the astrocyte, and it is transported, I can't remember the name of these, but it comes out of
glutamate, glutarase, and then it gets re-uptake by.
wait. Sorry, let me finish. it enters the pre-snap as glutamine. And then it is converted to glutamate by mitochondrial enzymes. Mitochondria, to lead into and then from here, it's intermediate, and then so, whatever not bound to one of these receptors, you get the uptake so here, as glutamate, and then convert it back into glutamine. Whoa, I'm not telling you. And this occurs via through some synthetic.
Did you say what terms glutamine and glutamate?
Yeah, they're a series of enzymatic enzymes or mitochondrial enzymes that are called.
What's glutamate?
The name doing specifically.
I believe glutaminase. Yeah, don't just know what terms one of the next. You should know at least then that you need to know the enzymes for it. It's not just this turns into this. You gotta know why. You won't need to. You're not gonna need to draw the organic chemistry structure and the reactions, but you do need to know the enzymes as well.
But like, I guess glutamate's also made as part of like the TCA cycle, right? Or it like there's like de novo glutamate synthesis too. So maybe that's what it's referring to
Yeah.
with the mitochondria, right?
Yeah, it's made from alpha ketoglutarate.
Yeah.
That's the intermediate.
And that's in the TCA cycle.
Correct. Did you guys have to draw the curve cycle in
I did not.
entirety?
No, and it's literally part of my thesis, so, no. But that doesn't mean you can't get asked, so can someone else go up and draw where the snare proteins are and explain what they do?
Oh, you got a case.
Okay. So the snare proteins would be located at the presynaptic terminal, these axon terminals, and there's different types. So you have your V snares, and you have your T snares, which is your tethered snares. And then you also have SNAP25, which is really important, and SNAP-decisive. So then, once your action potential comes down, the action and propagates and depolarizes, it will open these voltage-gated calcium channels, which allows the glutamate to be released in synaptic cleft. But how it is actually released in synaptic cleft is it needs to be synaptic terminals need to be tethered to the presynaptic terminal through the T snare. I can recall this very time.
Where are the T snares and the V snares? Are they both in the same area? Is one on the vesicle versus the membrane?
Yes, the V snares are on the vesicle and the T snares are on the membrane. What I remember properly.
Why does calcium flowing into the terminal cause vesicles to get released?
Why does it?
Why does it? What does calcium do?
Can I have a second to think about it?
You're missing a major protein involved in this process.
Yes, yes, there's also like. Anyone know the other protein?
Snaptobrevin.
Yes.
Yeah.
Yes.
So synaptobrevin is going to be a V snare. You're still missing one. Anyone? Yep.
any sense? And what is synaptotagmin specifically?
This one's the V snare.
It is a V snare,
Yes.
but what exactly is it for?
It's to help tether the synaptic vesicle to the membrane.
Yes, more specifically, it's the calcium sensor itself. And so that's why calcium, that's what triggers it.
That's what triggers, okay, that can release into the what?
Yeah.
Got it.
So all these V snares and T snares are gonna tether it, it's gonna dock to the vesicle, and it's gonna be fine, right? And then as soon as synaptotagmin senses the calcium, it's gonna be super rapid merging of the vesicular membrane with the cellular membrane, and that will lead to that vesicle pool. So remembering, like, synaptotagmin is the switch of the calcium sensor is gonna really, oh, that's a good way to think about it.
And could you say this is into the ready releasable pool?
So,
Just
yeah,
basically.
so there's gonna be a readily releasable pool at the, like, close to the active zone. And then, I'm trying to think about, like, the ones that are actually primed, right?
I don't think they're people.
Probably part of it, okay, yeah.
I would say there's vesicles that we call it primed and ready to go, we'll call it ready releasable pool. My understanding is ready releasable pool is any of the, like, vesicles that are basically just ready to go. And then the zone back, I can't remember I know there's the recycling rate for the ones that are just kind of waiting, but I can't remember the waiting area is still. Reserve pool, yeah, the reserve pool, and the reserve pool is back away from the active zone, anything that's basically in the active zone.
Can someone else go up and draw and explain the cascade of, a couple of proteins? Pick your favorite.
Yeah, pick your favorite metaphor or something.
You can try, but don't.
We'll help you out.
Yeah. So, just like any GI, GS, GQ,
Yeah.
So if you ask them which
like
one,
any, like a particular G protein, or
so if you ask them, we get to pick something hard. So if they just say something general, go with what you're most comfortable with.
Yeah,
Go with what
if
you
you,
know
if you,
best.
if you say which one, and they say G13, 14.
yeah.
So you can pick.
So, yeah, if they say to just draw one, then just pick one yourself.
So,
For something like that, it's usually they're going for of like your understanding of the technology, like they want to see like C-side, Q and stuff like that. They just want to see that you understand the words. From there, it could be quite a lot of things. And then another thing is you can start drawing. It's fine. We'll start like rolling off one another. So if you're studying dopamine and you mention a dopamine receptor, then they'll say, okay, draw the cascade downstream of that. Right? So kind of keeping in mind how all these flow.
Yeah, there's a very fine balance with the answers where you want to obviously say enough to answer the question thoroughly, but you don't want to start going off into all these random tangents where they'll just pin you into these rabbit holes that you don't really know the answer to. There's a very fine balance there. Because I have heard of people who will fail these because they're trying to keep their answers minimal to not give too much to go off of, and they fail because then they don't feel like they answered the question. So you do need to be thorough, but at the same time, just don't pigeonhole yourself into complicated that you don't understand.
okay, so, now I can't even pick, this is bad. Okay, so, I'll just do a, oh my gosh, now
Alright, GQ.
GQ. Okay, so for a GQ coupled GPCR, shit, now I'm blanking. Okay, so for a GQ coupled receptor, it would be, is that GQ, is it cyclic AMP or is this IP3? Yeah, okay, so
You're right.
for a GQ coupled GPCR, this would stimulate the G alpha subunit would go and it would simulate PKA, and then, or no, sorry, cyclic AMP. Oh god. Cyclic AMP, and then this would in turn activate protein kinase A, which then goes and phosphorylates all sorts of things.
That's GS.
That's GS.
Yes, yes.
Oh shoot.
GS is stimulation, right?
Yeah.
That's how I remember it.
GQ is IP3.
Yes.
Yeah. PHQ dice.
Phosphorus.
So yeah, GQ goes, the first thing it does is the alpha activates phospholipase C, PLC.
Okay.
And then from PLC, I believe the PLC then turns PIP2.
Well, does anyone know the GQ pathway? You wanna do it?
I can just try. I mean, so I think it has to do with the ER, right?
Don't you
So,
grabbing everything?
You have, it's G, for GQ, I think it's IP3, and then diacylglycerol, and then this leads to the release of calcium stores in the endoplasmic reticulum. Is this right? Or
So,
no? Or they're kind of-
diacylglycerol is gonna be in your membrane.
it's inside the membrane of the ER?
No, not of the ER, of the
Oh.
actual, the cellular membrane.
Oh, yeah.
So yeah, you might wanna start
Sorry.
with drawing kind of like the membrane, to show where things are. I'm also gonna be really nitpicky because I study GPCRs, so it's not what the GPCR looks like.
Do you want a 7?
Yeah.
Okay.
So you gotta have the 7.
3, 4, 5, 6, 7. I don't
Something
know.
like that,
Hey,
yeah.
something like that. Yeah, I mean, okay.
Yeah, also remember, again, this is a real nitpicky thing, but also remember that we also found membrane. And the things that they interact with first are also membrane bound. So like the PLC that this will interact with will be membrane bound, so they can be picky with that. So if you just randomly start drawing arrows into the cell when it's a membrane bound, then they may be on that.
And then what's the energy source?
This would be energy source.
So basically like when something
Is it
binds to
ATP? Or no, G, yeah.
GDP switches
G.
over to ATP. Once
Okay,
something
yeah.
binds to it.
GDP.
Labels what it's going from into. And you knew you had to just add that label, because I know you know it in your head, and you have like this mental image, but your committee won't know that you know it until you show them you know it, right? So, how it works out. So, you have your, DH's not there, right?
Oh, I
Yeah,
was
so
confused. I thought you said it's
so,
in the
yeah,
membrane.
so-
It is, it's in the cellular membrane.
Oh, I'm just saying like it's the thing that goes.
Yeah, IP3 does not stimulate like DAG, it's its own thing.
Okay, what is it?
So it's basically, so what you'll have here is, so I'm just gonna, so basically you have the alpha protein stimulating PLC in the membrane, and then PLC turns PIP2, and it turns PIP2 into both IP3 and DAG. IP3 is then the thing that goes to endoplasmic reticulum and helps release calcium from the ER to increase intracellular calcium.
Okay,
DAG
so,
goes off as its own thing.
so this is the next thing, right? Or it's like,
So tip 2 is like tethered into the cellular membrane.
this side.
Okay.
Okay.
So tip 2, yep, is like tethered there. And then your G alpha is gonna stimulate PLC, phospholipase C. In the membrane.
Yeah, PLC is also, is essentially.
Okay, cool.
Is PLC always attached to the membrane? Like, also, did it move? Because we were just looking at certain images.
know that it's, so, I don't know that it's in it, but this is where it's kind of happening in this pathway. So that I don't know, but just when we're talking about GQ, typically the G alpha is gonna stimulate proselytic C, so it's a lipase, right? So it's cleaving this lipid, which is, membrane bound. So it cleaves HIP2 into GAG, which is kind of the anchor. So that's gonna stick to the membrane. That's the part of the HIP2 that's in the membrane, GAG. Yep. And then IP3 is cleaved off of that. That's HIP2, and then that goes, yep,
Yes.
there you go, yeah. So is GQ, what is the effect of this on the cell?
It is stimulatory, or it leads to the signaling cascade that from calcium that can lead to depending on the context, but generally it's a stimulatory.
I was gonna ask, if you jump, so your GS pathway you drew, what are you missing at the start of the GS pathway? Because you're correct in that you have cyclic A and P and then PKA, but before cyclic A and P, what, there's a gap between the alpha and the cyclic A and P.
So how does the cyclic AMP get made?
Is it
Adenyl cyclase.
Yeah, so the G alpha will stimulate the non-cyclase and then the non-cyclase.
Is it adenyl cyclase is, oh, I see, so it'll stimulate this, and this actually.
And yes.
The cyclic AMP.
And then don't forget that the non-cyclase is going to be like that.
Okay.
Just like the
And then what about GI? Like, you don't have to draw anything extra for this.
So for GI, it's inhibitory for cyclic AMP, so I assume it inhibits adenyl cyclase, and then the net effect is reduced PK.
Yeah, I didn't know. So, yeah, those three pathways will be the main ones.
So let's weigh
Look,
it
if
as
Rick was on your
a,
committee, then
Yeah.
but so the GSGQ and GIO are definitely the three that you should know, because anyone could just get asked for like any of those. If you have any kind of research that might touch the G13, 14, if you wanna know what that is, know that one either, but it could, we don't like that it come up, I would think, if you just do it in general, if you don't really do this kind of stuff anyway, I'd be shocked if they do.
So if we do the alpha 13 steps, G13, 14, should we know all of them?
I don't know the pathway
All
myself.
of
I mean,
All of the pathways. If we use the alpha 13.
What, like, I guess what do you do? Is the receptor you study, it is the G, like,
Yeah.
G14, or are you studying specifically the pathways downstream of the receptor? Like,
Great question. So the receptor that I'm looking at, the alpha 13.
okay.
There.
And I would imagine that's not gonna be focused, if like the focus of your research is like the 13-14 pathway and stuff, then like, know it in and out, but if, like,
You should have a general.
yeah, definitely have a general sense.
Basically explain it like, surface level.
But there's like, how many different G alpha subunits, like, I think,
Yeah, no.
I hope no one would ask you that.
yeah.
Just something with Roe, that's all I know.
Do the
Active
most
Roe
basic one or something,
cytoskeleton
yeah.
remodeling, whatever that is.
That's what I was thinking, yeah.
Okay, so we just did GPCRs, what's the difference between the allotropic and ionotropic receptor? What are some main differences?
Direct conductance of ions through the pore of an ionotropic versus downstream effect on channels affecting depolarization and membrane polarity.
And I think the key thing with that is the speed of that, right? Yeah, so ionotropic is gonna be much faster, but in effect.
So what's the saying? Does anyone remember the saying?
What's the saying?
So, in long lasting. Yeah, slow acting, long lasting, fast acting.
I don't know, but just remember the one. So, what about, we have two ionotropic receptors up on the board, can anyone walk through the two different ones up there?
Walk through them in what way?
What are the differences between them and what happens when
Mm.
its ligand binds?
So we have AMPA and NMDA. AMPA
Wanna
binds
go.
to glutamate and to glycine to open a sodium channel, whereas NMDA has a magnesium that blocks the door. It requires binding of glutamate, opens it up, and then it's non-specific cation channel that lets in sodium, but it also lets in a lot of calcium.
And what about that magnesium?
It's a coincidence detector, so the postsynaptic membrane has to be polarized in order for the magnesium to block, so that ligand activity will actually cause conductance.
And then you said AMPA is a sodium channel, is that true?
No, depending on the subunit isoforms that configure the AMPA receptor, it can have different cation permeability.
Can you go into that a little bit more?
There are specific isoform subunit configurations that allow in small amounts of calcium as well as the sodium channel.
Do you know what the sodium
I'm thinking about that, I don't remember.
GluA2 is
Yeah.
QR and edited, and it's the Q version of GluA2 that is permeable to calcium over the R version. And I believe the Q is the one in the actual transcript, and then it's edited into R post-translationally, or post-transcriptionally, sorry. And then one small thing,
Yeah.
for the like NMDA, or you said AMPA was glutamate glycine, it's NMDA that's glutamate and glycine. AMPA is just glutamate, so it's the other way around.
And NMDA receptors require depolarization as well as the glutamate binding. And glycine is only needed in a laboratory setting experiment because in our human body we have enough glycine made, so we don't need glycine to activate. It's only when we put it into like a dish that we need both the coagonants of the glycine and the glutamate plus depolarization.
I will say, only because I used to work in an NMDA receptor lab, it's pretty complicated. There is like glycine and D-serine in the brain, so some people do think there are also like GluA3 channels that can come together with, like it's a, like the effect of like glycinergic and D-serinergic transmission in the brain is still like kind of like a debate. There's a, yeah, and there are other channels, but it's essentially correct. Yeah, you said.
What is true?
Yeah,
Okay.
yeah. I just, yeah,
That's why I thought.
just
Let's
nitpicky.
Who wants to drop action?
start labeling.
Well, so this was my first qual question, like it actually was
It,
asked.
it
Oh,
was
yeah,
like,
I got
it
it.
was like my second or third, yeah. You will be asked to draw an action person, so you should all be.
Whee!
Sorry, I wanted to only two people I can make eye contact with.
I didn't bring straws.
We can help you.
So you have time, milliseconds, and then you'd have voltage. Resting is at negative 70 millivolts.
Yes.
I would say around around -70, it's, yeah, not people are feeling very strong feelings.
So,
So. So you got an initial depolarization.
that, what you just drew that arrow to, that little part that goes down, is that depolarization?
No, that's hyperpolarization.
Apple.
Which, is, I mean, it still fits technically speaking, because you can easily have hyperpolarization before it gets to depolarization. Yeah.
So you have stimulation causing depolarization until you hit the threshold potential. That causes opening of sodium channels, which causes a rapid depolarization. Then you get a refractory opening of calcium channels, or potassium channels, sorry, as your sodium's closed.
What?
So which direction are those, ions moving in and out of the cell?
That's in. Well, no,
Does sodium move in and out of the cell during,
sodium goes in, potassium goes out.
Yes, that's good. I'm curious what you mentioned potassium there, you said that there's a refractory opening of potassium channels? What do you
Well,
mean
I
by
just
that?
meant, in response to the change of polarity, the sodium opens. The refractory period is that the sodium channels have a time duration until they can become activated again. So then you have the potassium close, leading to polarization and then a return to resting membrane. And then you have the refractory period.
I don't know if this is an official term, but would you say they close or they're leaky?
Well, they close at different rates.
Right.
Yeah,
So different isoforms have different sensitivities.
There's a lot of different types of potassium channels.
Yes.
so then why do the potassium channels open later compared to the sodium?
Delay rectify. Well, so they're voltage sensitive. So because I'll say dynamics within the membrane pore, there's a voltage sensor which kicks on when it reaches a certain depolarized voltage. And then you also get a switch in the Nernst potential of ion conductance relative to membrane potential.
So what does the Nernst equation tell us?
The Nernst equation tells you ion flux of multiple ion species given a particular membrane polarity.
Can you write it out?
And that is, it sounds ridiculous, but that is a fair question, right?
Yeah,
Yeah.
Nernst. It's
How about the gold medication? This is an example of when you mention something and then it
Yep.
reminds a committee member, oh, well, what about this, since you've mentioned it, right?
I think other people in the room can help them too, if you
Yeah.
have it.
So I think then my understanding the Nernst is just one ion species, and then it's the GHK that's
GHK,
all of them
multiple.
together.
Yeah.
And I think Nernst, you need, it's like RT over, like, I think R and T is like some constant. T's the
Yeah.
temperature. What's R?
Gas? No,
I don't know. And then it's over. There's the
sorry, gas.
yeah. That.
So,
God,
Well, now I know it.
so kind of feeding on this, so why does sodium go in, why does sodium come into the cell, and why does potassium leave the cell?
Because they have their own electrochemical gradient based on the concentration inside and outside of the cell, which is maintained by ATP dependent sodium potassium
What is the Nernst equation, that E, what does that E stand for? What is that telling you about that ion? What is that number? Yeah, that's probably likely too, is like, the reason sodium testing go in and out is trying to reach those respective people again.
Which is not only dictated by inside outside, but also it's electric gradient.
So you just mentioned the sodium pump. Can anyone go up and draw what it does, where it is in the cell, what it's doing?
Ari, would you like to type out what a sodium potassium tank is? Can
No.
you describe it? You don't have to draw it, but if you want to know what it is.
I think you said no. I mean, I don't know, I don't know the answer, but I can go up and embarrass myself again. But I'm pretty sure, like, for the sodium potassium pump, it requires like active transport to like your ATP in order to, I don't know how many sodium ions are and potassium ions are actually exchanged.
I think it's 3 and 2.
3 and 2, so this would
3
be
sodium out to 2 potassium in.
3 sodium out. Okay, so you have your sodium in.
So Na, and an arrow out. Right?
Different.
There you go.
Two.
That's right.
Ta-da.
Virus.
Baby bubble.
So, something that I learned, I think, in literally middle school that I still use as neuroscientist is salty banana. I don't know if anyone's heard this before, but it's a salty banana. A neuron is a salty banana because there's cells on the outside and you can't see on the inside. And that is literally still the only way I remember it, so.
So you mentioned the refractory periods, refractory period at the end. Can you go more into detail about that?
In order to rectify the hyperpolarization of an action potential, you have to activate ADP hydrolysis to renormalize the ionic concentrations.
So could you fire an action potential again during the refractory period?
No.
Isn't part of the refractory period?
How many
the-
refractory periods are there? Or how many stages are there?
How many stages of a refractory period there is?
There's two,
Yeah.
there's relative and absolute. Absolute is the part where, so what makes the absolute refractory period the absolute refractory period compared to the relative?
Well, so it's the inactivation of the sodium channel itself with the like ball and chain thing where it like closes itself.
Yes.
And then it needs to like release that in order to
Yep.
actually pass active.
Yeah, when the sodium gates are still inactivated, no amount of depolarization will cause them to open whatsoever. That's absolute. As soon as the inactivation releases itself, you go into relative. And in relative, you'll still be below the resting, so it's harder to get to the threshold, but you can do it. So, yes.
That being said, can anyone explain to us burst firing? What is burst firing?
I guess it'd be like a train of action potentials in like rapid succession in response to like a single like over threshold or single depol or like you depolarize the membrane enough so that it reaches the activation threshold and then you get a train of action potentials like immediately after that.
So what would that look like on a graph? Burst firing, if you were measuring it.
You want me to try it, or?
Yep.
So it'd be kind of like this, except you could go over the threshold, and then you would get a bunch of these. Yeah.
Yes, that's perfect.
Why it happens? Certain cells do it. Everyone knows.
You're gonna talk about LTP at all?
Yeah, let's go into LTP and L.
We'll start. So, yeah, does anyone want to draw LTV?
What does it stand for?
Long-term detention agent.
I mean,
Long-term
they're like
depression.
short-term, like shorter term, longer term. That's like chemical versus structural. So the kind of like physical transplants are just more likely to get neurofiring, but the structural ones like actually have changes to like dendritic spines and such.
Can you draw it out what you're saying? Like what you would have to for the drawing too?
You got this, baby. Do it.
I
I've already embarrassed myself too much. It can't be any worse than my GPCR pathways.
I guess. We stop here, and then if we stimulate it a lot, you might get neurons like neurons respond with like more receptors being activated in the membrane. So when the same amount of stimulation where like transmitter gets released to the same spot, there's just like stronger downstream effect because there are more receptors. Or if that keeps happening, you get long-term potentiation where, imagine in dendrite, they used to have like a couple of spines maybe, and now it could have like crazy, I don't know, lots of spines or structural changes.
So you hit a couple ways that a synapse can become stronger. There are a couple more. Anyone?
Phosphorylation receptors.
So more active receptors, right? So receptors are gonna pass through more current or okay, and then
There's also like presynaptic LTP, right? So more vesicles released.
and then they can also become bigger, right? What's the key like molecule in this LTP process? What molecule is like that?
Calcium.
Yeah,
Yeah.
exactly. And that's that magnesium block thing we talked about earlier, right? Once that magnesium block, it's like the coincidence, and that's what allows the calcium, which allows the LTP. So ketamine will tie those two things together.
And then what does calcium do? Get some numbers. How does calcium, what does it activate?
Calcium binds calmodulin, which then activates camp kinase 2, which phosphorylates AMP receptors. I believe.
I believe the CAM kinase does a lot of things.
Yeah.
That's one thing. I think the last one was magnesium crab.
Yeah.
So if you know from calcium to calmodulin to CAM kinase 2, that should be sufficient for that pathway for us to draw.
Yeah.
Is everyone following what's being said right now? Like those who are not talking? Cause this is a very common question you'll get on your wall. And so if you want someone to go up and draw it and explain it for your benefit as well, not only does it help them, but it helps you. But speak out if it's not making sense or doesn't ring a bell or sound familiar.
So like what level of detail Do you think we'll need to know, like, is that sufficient, or do we have to know, like, 4 calciums bind to calmodulin, and then you get,
Well, not
Like-
that specific.
okay.
It's not from my,
Only
it's
if
not
they
my.
ask you, and it's okay if they ask you, let's say you've answered the surface level question of drawing an actual potential, and then they continue asking for more and more detail. They're trying to get an understanding of where your limits are. So you will hit a point where you don't know an answer, and that's supposed to happen. But when that happens, you can try to give it your best shot. They wanna see that you're at least attempting to answer it, or come up with an experiment to figure it out. So,
I will say, yeah, like my project has absolutely nothing to do with me, and everything we talked about so far, I was asked.
Oh, that's the biggest.
Like in much more detail and more, and like I was even asked questions about the anti-NMDA receptors and what anchoring proteins on the postsynaptic membrane hold those, and like that was where I was like, I don't know.
Homer Shane.
Yeah, and Bruce was like, all right, like I feel like at this point, like Rebecca shouldn't have to know that, considering like it has nothing to do with my project, but if it does, like we need to be asked that, and that's fair game.
If anyone is an electrophysiologist, be able to show how you would measure all of the things that we talked about today.
Yeah,
Even if you're not
also-
an electrophysiologist.
I'm also not an electrophysiologist, and I was asked that, so I would say no, just no different methods of what you can do, and what you can measure, and what data you can get from those things. and limitations of them. I have, like, I don't do any of this, and I was asked all of these things.
what were you about to say? Oh, just that my PI does, but that doesn't matter. Yeah. If your committee members know your research, they might be interested in getting an understanding of your knowledge on the lab research, not just your own. So you might still be asked those questions.
I think EFIS is definitely fair game for anyone.
Eva.
Yeah, the depth of EFIS that you need to know is gonna be dependent on who's on your committee and stuff, and who's your PI.
Yeah, if any of the people on your committee do EFIS as a thing, then you'll need to know it, like say beyond, a little bit more than surface level. You need to actually know a little bit of detail.
So can anyone tell us about patch time? And the different things you can do with it? Different test different questions.
I could, but I don't want to monopolize. Go
No,
ahead.
no, no, I actually still had a question about LGC. So as we were going through everything, it kind of, like, I understand it, but it got a little, it can get messy and it can get messy quick. So, maybe the best approach, and if one of you would be so kind, is like, if you were asked this on your call, how did you answer it? Like, for me, how I'm thinking of going about it is doing basic level stuff first. Like, if I just draw an action potential, I would just draw the action potential, and then start asking questions.
You should
But-
have some labels on there.
Well, labeling, but basic, but
Yeah.
not to
I will say.
start at the bottom of a rabbit hole already.
Yeah, so
Does
you
that make
likely
sense?
get asked like the synapse questions first. And for me personally, like, I started to draw the synapse and then Bruce stopped, he was my program rep, and he stopped and was like, can you make it bigger, because we're gonna be asking
Okay.
more questions, and like, you're gonna be drawing in it, so, so I had to like make it bigger. So like I started drawing the synapse, and then when we started asking questions about, you know, calcium and, going more in depth with the LPP, I just drew that into the postsynaptic membrane, and then I was like, okay, then we're adding more receptors, so I had more room on my membrane to add receptors and like LPP, or if you need to add, you know, like, just make sure you have enough room, like, right now, it's kind of messy up there, so you wouldn't have enough room, but like, he made me draw, like, my synapse was probably like a third of the size of the board, like, just draw bigger.
In terms of erasing things to make more room, ask them, are we all set with this, can I erase? Something, like today, for example, what we're doing is, I would change the topic, but then someone else could go back to something we had already discussed and be like, okay, so, like, kind of like when it's their turn, they wanna go back to something you thought you finished up, but you're not quite done with yet, so before erasing anything on the board, ask first. And, they might ask you, okay, we're done with this, go ahead and erase it, right? So, yeah, get in the habit of leaving things on the board. And I think it's fine to approach it by like starting with basic, but you'll gauge a sense of whether or not your committee is okay with that. If they seem like okay waiting for more, keep going. If you turn around, you're done drawing, and then they ask a question, then you're good, right? But if they're waiting for more, I would suggest keeping on going, even if you think you're gonna start with the rabbit hole. That's what they're expecting.
Let's say like, I think the action potential one was a good example of how to, like the appropriate depth. I mean, it's all subjective, so I can't speak for everyone, but like, I think drawing the action potential and saying, okay, this phase is what, like, this is what's happening here, this is what's happening here, this is what's happening here, and it's because these are goals indicated, right? So kind of just saying, not like, when there's the alpha subunit, it's at this, like, you know what I mean, you don't have to go into that, but still like giving enough information, not just like drawing the curve and being like, that's it.
Right.
But like, this is what's happening at each phase, this is generally what's happening inside the cell, maybe going into like, depending on like where if it propagates, like then you can just tell what was left.
So I'm still trying to just like wrapping my head about how few details you can start off with. And so, like, say we were to do like the action potential question again, and you were just to write, okay, this side is deep hole, this side is hyperpool, this is where the refractory period occurs, like, without including the things about the sodium and potassium channels up there. Would that still be like a substantial place to start and let them ask you?
Yeah, you don't have to include the sodium and potassium
Okay.
channels. I don't think so.
I will
Unless
say,
you're asked.
so my first question was an action potential, and I went into it, because I was like, I'm comfortable with this question, and I went into it, and basically, like, to the point where the committee member was like, okay, that's enough.
That's actually.
Like,
exactly what
they're like, you've proven that you know this, and then going forward, they're like, okay,
Please.
they're like, kind of like, unite shit. So if there's something you feel comfortable with, I would show it. Again, I wouldn't start talking about like subunits on there, but, I think if you're starting off with something you're comfortable with, just show that you're comfortable, show like getting off on a good foot to begin with is gonna be really important. Or I should say like, that's what you want, like, if you don't, you still have, like, you're gonna be fine too, but like, if you're able to, like, that's a good way to get started. And that's why the 5-minute talks are helpful too.
So we're almost out of time. Are there any like last minute questions for our experts for today or?
You guys asked Beth about LTV, long-term regression.
I might have been, LTP was a big one for me. I was just straight up asked, like, draw the mechanisms of LTP.
Okay.
And on all of them.
Mechanisms.
LTD might have been like a tack on at the end for me,
Okay.
but I would assume, like, LTP is probably more important overall, but I would still know,
Are
like,
you
some of the mechanisms of LT, maybe it's not, I don't know if you need to know quite as in depth.
Okay.
That's what I would say.
I would know the difference in what type of manipulation causes each one.
That-
So like, when would LTP happen versus when would LTP happen?
They're the two different like calcium binding. I can't remember it,
Like
but it's,
Creb and
there's calmodulin and then there's the other. It's like
AKAP, I think, binds
like,
calmodulin
is it- I thought it
and
was like
does Creb.
I thought it was neuro something, but
or Cadget.
Calmodulin.
there's another one, right? And it's like lower
Calnurin
affinity
or
for
something?
calcium, and so
Oh,
because
calcium.
calcineurin,
Calcineurin.
yeah.
Calcineurin.
Yeah, there's calmodulin, there's calcineurin, right? And like one's high. I don't know. Should we know?
That's actually, yeah, honestly, yeah, I was calling you out, because I think you said one's more NLP specific, one's more LTP specific.
Yeah, because like one of them, like in LTD, I think it's a calcineurin because it's like low calcium stimulate, like it's low levels of calcium because it's high affinity for calcium. I think it's calcineurin. That one gets activated and then that leads to LTD somehow, question mark. And then calmodulin is like low affinity for calcium. So
Is that more downstream?
it like needs high levels. I think it just needs high levels of calcium in order for calmodulin to I don't know.
It's one of them, because I think LTD
Yeah.
is low amplitude, but with a frequency around 100 hertz. So like high frequency, low amplitude, whereas LTP is higher amplitude but lower frequency. I'm not sure if that's true.
Say that again?
What
The
frequencies?
amplitude and frequency dictates whether it's LTP or LTD. It's a train of 100 hertz induces LTD, whereas a paired pulse, you could do paired pulse facilitation.
I remember that the frequency does have something to do with it, but I don't know if it's a distinction between LTP and LTD, but I think there is like a threshold. Don't quote me on that, I would go check, I don't remember, but
I also don't know the answer, but it would be really easy to look up because there are specific protocols that we, the electrophysiologists, use to induce either one.
I would say also what's fair game is, if you're asked to design an experiment around this, knowing key drugs related to this. So like ketamine's a big one that's talked about a lot, which is an NMDA4 blocker, who kind of beat the hell out of it in a bunch of different classes. So like, I would just know key drugs if you're asked to design an experiment where you need to, need some sort of inhibitor or some sort of blockage, whatever, know those. You might get asked, like, common ones.
I would also, because this is something that came up that kind of caught me with surprise on my own, was, so I was talking about LTP and like how you measure it, which you can record from the cells and see differences in their, potentials. There's also a question which is like, how do you measure like presynaptic, like how would you measure like presynaptic like function? Like if you have cells that go through like changes or whatnot and they change, but it's on the presynaptic, so how do you measure presynaptic function? Just the main thing for that.
Be looking at frequency of ESCs, right? Versus like amplitude. Because that'd be like the amount of events released, right?
That is an answer.
It is, yeah, I was looking for
Are you looking for paired pulse facilitation or?
pair pulse facilitation question. Pair pulse ratio as well.
You might answer a question right, and they might say, that's exactly what I was thinking of. And then you have to play like the Miami game.
Aerial Dory, because that's his.
Oh, he's not.
Nice.
Does that mean we're all passive?
I assure you. But yeah, because I think a lot of us have had an experience where someone wanted a very specific answer, and it's really hard to get to what they're really talking about. Hopefully, people on your committee will be like, that's a good enough answer. But if not, feel free to ask them for a more specific question.
Right, I did that a lot. I asked a lot of clarifying questions, because, they might ask a broad question where you could go like 9 different directions in answering it, and to ask a specific, right, what do you want me to draw here?
Yeah, and it could work in your benefit, right, where they're not looking for anything specific, kind of like when we said go draw a synapse, we're not thinking of anything specific, but it could also not work to your benefit if they are thinking of something very specific, so, you'll get a sense of what's going on after you've given your initial answer, whether they're satisfied with it or they're looking for something more.
Talk to people who have had the same thing.
I think we're out of time. I sent everyone a spreadsheet, to populate as you guys get your committee confirmed and your qualities confirmed. We're gonna try to schedule your mock calls around, based off of whenever your actual call is happening. So, that should be in your email now.
And if you have any questions, you're always welcome to email us.
Real quick,